Hello, my name is Guy Young, and I'm very pleased to give you this short presentation about gene therapy and hemophilia, update on efficacy in clinical trials. As the title indicates, I'm going to really be discussing only efficacy. I won't be discussing the other issues, of which there are many, revolving around gene therapy. So we're going to get into the details of the recently published clinical trial data. So here's what I'm going to review. We're going to talk about Factor IX first, and we're going to look at two products essentially that are in clinical development. And then after that, we'll discuss Factor VIII, where we'll review data from 3 different products. Okay, so let's take a look first at gene therapy with Factor IX, as we're planning on discussing. And here we're looking at gene therapy with an AAV5 human Factor IX vector. And this product is known, or was known, still known, as BMN270. I say was because it is no longer in clinical development, and we'll discuss that momentarily. I think the important thing about this trial is you see that you've got data in 2 cohorts. These are 2 different dose cohorts, and the data is out about a year, slightly more for some patients in cohort 1, and about 6 months in cohort 2. What you can see is that the factor levels are hovering in cohort 1 between about 1% for one patient, but mostly between about 3 to 8%. In cohort 2, you see that the levels are a bit higher, going from about 2% up to as high as 12%. I think an important part of this data is you see that the levels are very stable, that for each individual patient, especially if you look at cohort 1, which has 1 year's worth of data, you see that the levels are remaining very stable over time from patient to patient. I think that's the strength of this data from this phase 1 study, is that you've got steady levels over a long period of time. Now, taking a look at the issues related to anti-AAV5 neutralizing antibodies, what we know from many of the studies that are going on is that patients with AAV5 or AAV neutralizing antibodies, regardless of the AAV, often are not included as participants in those trials. In other words, they're screened and they're screened out for participation. But in this trial of AMT-060, those patients with AAV5 neutralizing antibodies were not screened out. They were tested, but they were not screened out. What you see in this figure here on the y-axis is the antibody titer, and you can see that's a log scale. And on the lower axis, you can also see the antibody titer using a different method. So you've got the neutralizing antibody titer using the luciferase assay compared with the ELISA assay. And you see a nice correlation between those two. In other words, the higher you are in one assay, the higher you are in another assay. What's important to point out here is looking at the individual patients and their factor levels, is what you'll see is that there are patients like patient 4 who happens to have the highest antibody levels, regardless of which assay you look at, but he also had the highest Factor IX level at 7.7% in this group. And so looking at this data, it suggests that there really isn't a correlation between the neutralizing antibody level and the achieved Factor IX level. And so this suggested that for this particular vector, this AAV5 vector used in this particular gene therapy product for Factor IX, that neutralizing antibodies did not correlate with the Factor IX activity. And that's why moving forward in this particular program, the plan was not to screen patients out. In other words, not to eliminate patients who screen positive for the AAV5 neutralizing antibodies. So looking further into this cohort of patients, again, cohort 1, the lower dose, cohort 2, the higher dose, we're looking here, again, Factor IX activity. And now we're looking at a further analysis, which was published recently at the last American Society of Hematology meeting. And now you can see that data is going out, getting beyond even 3 years. And you continue to see that for cohort 1 at 3 years, and even cohort 2 at 3 years, that the Factor IX levels are remaining stable for each individual patient. Yes, the levels are a bit low, and we'll get to the newer product that is being developed from the same essential overall study or the same company and the same vector, and we'll show you that the levels can be made to be higher. But the main message here is that steady levels can be achieved with Factor IX over a period of 3 years. And again, importantly, that these patients, some of them did have positive neutralizing antibodies to AAV5 and yet still remained with stable expression over time. So moving forward to the next phase of development here is that the company has decided that those levels of Factor IX, it's not just the company, I think all of us in hemophilia are at the point now where a factor expression level of 4, 5, 6, 7% is no longer considered really adequate. What we really want is higher levels. levels. And so what was done here is that the same AAV5 capsid that I just showed you in the AMT-060 trials, the one that gave you stable expression and the one that did not require patients with neutralizing antibodies to be screened out of the study, was then being used with just a slight modification of the transgene, which is to change from wild type factor IX, which is AMT-060, as you see there on the right, to the Padua variant of factor IX, which has a 6 to 7-fold increase in activity. And we're going to call that product now AMT-061. So everything else is the same. The only difference is that the factor IX gene now is the Padua variant. And so here's data published again very recently from the phase 2b study. So what was done was 3 patients participated in this phase 2b study. And you can see here their factor IX levels out 6 months. Again, same vector as before, just a different transgene with Padua. And you see that the mean factor IX activity here, as expressed by the one-stage clotting assay, is 47%. And you see the 3 patients, one at 33% and the other 2 at 51 and 57%. And they're fairly steady once you hit their peak at around 4 weeks or 8 weeks. They are remaining fairly steady. There's a bit of an increase in the top 2 patients, even out to 6 months. But all in all, you're seeing fairly steady levels. So this is the product that is moving ahead into the phase 3 study. And that data we're still waiting to see that data. So the next product actually is different. This is briefly data that was presented at the EHAD meeting, the BMN270 study. So this is, again, a factor IX Padua transgene, but the vector here is called AAVS3. This vector is designed to be more efficient at transfecting hepatocytes. And therefore, the idea here is that you'll have better hepatocyte transfection along with the factor IX Padua transgene, which in theory should give you even higher levels of factor IX. So here's the data from that phase 1 study, and I'll show you a couple of slides on that. So first of all, what you'll notice here's dose level 1. It's a fairly low dose. It's 4.5 times 10 to the 11th vector genomes per kilogram. The other programs are using doses more in the range of 1 times 10 to the 13th or in the 10 to the 13th range. Here we're in the 10 to the 11th range. And what you'll see for this patient, you can see the factor IX activity, is that going from week 3 out to beyond a year, week 78, you see expression that is not quite in the normal range, but getting close to the normal range, albeit with really quite a low dose of the gene therapy. And this patient did receive prophylactic steroids, so that was the strategy to prevent any immune reaction, was to use prophylactic steroids, and there's the factor IX levels as you see there. So here's the second patient. This was the higher dose level now, 1.5 times 10 to the 12th. So it's about half a log higher than the last dose. As you can see, the dose level 1 still there. And here you can see there were 2 patients who participated, again, using prophylactic steroids. And here's the factor IX activity. And now you can see factor IX activity getting well into the normal range, even at week 26 above the normal range. These patients did not bleed, and really this is suggesting that this type of vector, this higher transfecting vector, is really in fact having its intended effect. In addition, tacrolimus was being used for the first time to control breakthrough elevations in the transaminases. So the patient were on prophylactic steroids. However, at this dose, there was some elevated transaminases, which seen with loss of expression, and therefore tacrolimus was added. Moving now to the third dose level. So now you'll notice that this dose level is a step down from the last one, right? So dose level 1 was lower, went up to dose level 2, and dose level 3 is stepping down. It's in between the other 2 doses at 7.5 times 10 to the 11th vector genomes per kilogram. And again, prophylactic corticosteroids were used, tacrolimus for breakthrough elevations. And what you'll see here is that the factor IX levels now did not achieve or get into the normal range, which ultimately was the goal. And getting finally to the last dose level, which is a little bit higher than the third dose level, not as high as the second dose level. So you can see this strategy is really trying to find the sweet spot. So this dose is 9.75 times 10 to the 11th vector genomes per kilogram. So just about 1 times 10 to the 12th. And there's the 2 patients. Now these patients received prophylactic corticosteroids and prophylactic tacrolimus. And that's because in the previous 2 dose cohorts, there was some loss of activity, even using prophylactic steroids and reactive tacrolimus. So tacrolimus was added as prophylaxis. So here you can see the mean factor IX activity is right there in the middle of the normal range at 109%. And so this is the dose that is going to move forward into further trials with that product. Okay, so those are the two factor IX products that we have recent data on. I'm aware that there are other factor IX gene therapies, however, they haven't presented or published data recently. So let's focus now on factor VIII, and we're going to look at three products: BMN270, SPK8011, and SB525. You can see the companies that are the sponsors or manufacturers of those products. So first, we're going to look at the recently published data from the multi-year follow-up of the AAV5 human factor VIII SQ gene therapy. This was known as BMN270. It is now called by its generic name, which is hard to pronounce, valoctocogene roxaparvovec. We prefer to call it Valrox, so that's kind of what I'll refer to this product as going forward, is Valrox. So what I want to show you is a few different patient profiles from this study, and I'll walk you through this. So here's the first patient. So first of all, what you should note is that the factor VIII activity is in the green histograms near the bottom there. At the very bottom is the dose of steroids. You can see that this patient started steroids early, was receiving prophylactic steroids. And you can see that as the steroids were tapered, the transaminase levels went up. You can see that in the yellow colored dots, which is the level of the ALT. As the ALT went up, the prophylactic steroid dose, which was being tapered, was increased. Then it was tapered again, but the level of transaminases went up again. And what you can see is that this patient really has quite a low level of factor VIII expression of around 5%. And then you see over time out to about 3 years, it's actually quite low. It's about 2%. So, and you see at the top there, those are bleeding events in the dots, as well as factor VIII use, both one-time use and use for surgery. So here's a patient who did not have a really good response. And part of the lack of a good response may be related to his immune reaction as his transaminases went up. And you can see that while he didn't have much expression even at the beginning, that his expression was lost. Yet here's a different patient. You can see that this patient has achieved rather quickly normal factor VIII levels, again, represented by the green bars, factor levels reaching 80% and even by almost at a year at about 100, 120%. You could see this patient also was on prophylactic steroids. They were tapered rather quickly, going from week 2 out to week 16. And you see that there was really essentially no transaminase elevation. There was maybe one slight blip at about 1 year, but for the most part, the transaminases remained low. And you can see this patient out 3 years has a factor VIII level of about 80%. So clearly that patient is doing well. Remember that the dots at the top are factor usage. You can see that there, other than on day 0, there are no dots at the top. So this patient is not bleeding and is not using any factor VIII. And then here's a patient who's sort of in between. So this patient needed a longer course of prophylactic steroids, had some minor elevations in the transaminases, did achieve a level up to 80%, but then over time, that level has dropped. You can see out at 3 years that this patient's level is around 15% of factor VIII, with no further transaminase elevations and no further use of prednisone. So what we've learned from these 3 patients is that there are some patients with not a good response, there are patients with an excellent response, and patients in between. So the main point here is that the responses are really quite variable. Now, if we take the whole group as a whole and look at their factor VIII activity over time, and now you can see here 3-year data, what I'm showing here is in the green bars is the chromogenic factor VIII assay, and in the yellow bar is the one-stage assay. And I'm keeping this here, or I kept this in this presentation to illustrate the point that the one-stage assay is about 1.6-fold higher than the chromogenic assay. Now, this company is moving forward with application to the Food and Drug Administration to get this product licensed, and they're moving forward using only the chromogenic assay, as they believe that this is probably more accurately reflective of their data, or at least that they're taking just a more conservative angle and using the lower levels. So it's important to note that, if and when this becomes available and you're using it, that keep in mind that the one-stage assay is going to give you about a 1.5 to 1.6-fold higher level. The other thing to note from this data is that patients peak at around 6 months, and that there's a slow drop-off. You can see a drop-off by week 52. The means and medians, as reflected by the diamonds and the dashed lines in this box and whisker plot, are decreasing. So from week 24 to week 52, there's a slight drop, and then a further drop from week 52 to week 104, and then a continued further drop from week 104 to week 156. I will say that the 4-year data was recently presented at World Federation of Hemophilia, and that level has continued to drop, albeit slowly. It is further decreasing. And so what we know from this product is that patients can achieve normal factor VIII levels and that over time there's a slow, steady decrease in those factor levels. But yet, even here at 3 years, you can see that these patients have levels, a median of 20%, a mean of around 28% in the chromogenic assay. And again, the chromogenic assay is the one that's going to be carried forward in the phase 3 trial and the one that's going to be used for the biologic license application that is under review by the Food and Drug Administration. Now, importantly, we should also look at bleed data and factor usage. So you can see here annualized bleed rate on the left, factor usage on the right. You see before infusion, you see in this case the annualized bleeding rate was about 16 for an ABR of 16. And there you can see the reduction in bleeding. The two bars represent two different dose groups, but basically you see that there's a similar ABR before. And then when you look afterwards, you see there's a 96 reduction in mean annualized bleeding rate, essentially going almost to zero. And commensurate with that on the right, you see a 96% reduction in mean factor VIII usage, which makes sense, of course. If you're not bleeding, then you shouldn't be using factor VIII. Okay, so let's move on to the other product. This is SPK8011. What I'm showing you here is data from actually late 2018, so it's about a year and a half old. I'm aware that just last week, as I present this to you here, that Spark presented additional data on these patients that is out now 2 years. Suffice it to say that some differences you see here. First of all, it's a small number of patients. That's number one. Number two, you see the dose here. The dose is in the 10 to the 11th and low 10 to the 12th range. So it's about 1.5 logs to as much as 2 logs lower dosing. And then you can see in the dashed red line, what I put in there is roughly where the factor VIII level is falling, which is around, mostly 10 to 15%, although there's one patient that sort of inexplicably rose after 24 weeks. The 2-year data that was just recently presented, and frankly didn't have enough time to get it into this presentation, shows steady levels out to 2 years. So it's a little bit different than what we saw with Valrox. There are steady levels, albeit the levels are also a bit lower. Some of these patients did need corticosteroids, and some of the patients needed corticosteroids for quite a long period of time. So I think what we can say from SPK8001 is that we have much less data, fewer numbers of patients, shorter duration, and yet their lower doses of the gene therapy product are leading to, generally speaking, lower factor VIII levels. The question is, how will this move into phase 3? And I think that that is yet to be determined. What will be the dose for phase 3? How will the corticosteroids be managed? And I think only then can we make a fair comparison of this product to the BMN270 or Valrox that I just showed you. So really, this is just to let you know that there's another factor VIII product that is for gene therapy that is in development, although data is still a little too early to really pass a lot of judgment on this one. But ongoing development will lead to more data, and then we can make more rational conclusions about how this product compares to Valrox. And then finally, the third product is the SB525 product. This one is even further behind, so you see even fewer data. You just see 4 subjects here with follow-up out to about 6 months, 25 weeks. This was presented about a year ago at ISTH. Yes, they did present some additional follow-up data on this group of patients, but again, the follow-up is still fairly short compared to Valrox, and the number of patients is fairly short as well. A couple of things you'll notice is that the dose here is similar to the Valrox dose, which is 6E13. You notice the dose here is 3E13. The other thing you'll notice is that for the patients that are furthest along, you see 2 of them there, are getting into the normal factor VIII range. But again, this is about where we saw Valrox as well at 25 weeks, or roughly 6 months. So it's too early to tell whether or not these levels will be maintained in these patients. I think it's just something that we're going to have to wait to see further data and see if we see the same effect of a drop-off in the factor VIII levels. So I think all we can say at this point about factor VIII gene therapy is that we have 3 products, one that we have 3-year and even 4-year data on, others that we have 1 to 2-year data on. And for the product that's Valrox that has been out the longest, we are seeing a slow but steady drop in the factor VIII levels over time, although those patients nevertheless are hardly bleeding or hardly using factor. For the other 2 products, there's somewhat different approaches, with Spark using a lower dose, this SB525 using a similar dose to Valrox, but the data isn't mature enough for us to be making comparisons. So with that, I'm going to conclude by saying that there are multiple gene therapy clinical trials underway. They're all using the AAV platform. There are preclinical studies that are assessing other approaches, including other viral vectors such as lentiviruses, non-viral gene transfer, and gene editing. And that one product, valoctocogene roxaparvovec, may be approved as early as August of 2020 in the United States, and we'll have to wait to see what the FDA rules on that. But I think we're entering an era of really exciting times where we are going to have, in all likelihood, gene therapy as an option for patients, both for hemophilia B and for hemophilia A going forward. Although I have to say that there's still a lot of unknowns. The unknowns include overcoming the immune reactions, whether or not we need to use immunosuppression for a short term of time, long term of time, prophylactically or reactively. Durability, clearly with factor VIII, there are certainly some questions about durability. Factor IX, the durability seems to be a little bit more robust. Also long-term toxicity, including insertional mutagenesis. And then obviously there's going to be issues about access and cost, and then implementation. Implementation meaning that this is a genetically modified organism, and it's not so simple to just bring it to your pharmacy and infuse it. There are certainly issues that centers are going to have to understand about how to bring this product into their center, how to do the infusion, and how to do this safely. It's not like bringing a factor product and infusing factor. And then lastly, what about the use in young children who arguably be the ones who most benefit from gene therapy? I think there's a lot of interesting questions we have to ask there, and a lot of work that needs to be done before we can consider these types of therapies for young children. So with that, I'm going to essentially close by using this very nice graphic from Glenn Pierce and Alfonso Iorio about the past, present, and future of hemophilia gene therapy. They talk about having known knowns, which are the things that we know that we know. So some of the data I presented. There are things that we know that we don't know. For example, the long-term safety effects. So we know that we don't know the long-term because it's going to take time to develop that. And then there are unknown knowns, in other words, what we forgot that we know. And then, of course, the unknown unknowns, things that we can't even anticipate that may happen in the future. And so I think for our field, I think continuing to collect data, continuing to collect long-term data on what's happening with these patients over time. And there is a planned World Federation of Hemophilia registry that will enroll hopefully all the patients who get gene therapy into long-term data collection tools, registries, so we can really make sure that we're bringing this to patients in the smartest and safest way that we can. And so with that, I will close, and thank you for listening.